Hello, my name is Dr. Danny Meyersfeld. I am the founder and CEO of D-Analysis Biotechnology, the partner company in our international collaboration with Nordic labs called DNA Life. For the ensuing lecture series, you're going to be hearing a lot of new terms, new language, new terminology, many concepts that you may not have come across before or that you may have come across but not in the recent past. So what I'm going to do today is start off by reminding you of some of the basic concepts around DNA, some of the biochemistry of what happens in our cells, and some terminology that is essential to ensure your understanding as we progress through the future lecture series. So to start off with, I'd like to introduce this molecule to you. I'm sure it looks familiar from a few years back when you were in high school studying your biology. This is one of the most recognizable structures in biology, the double-stranded helix that makes up our DNA, our hereditary material. The complete human genome, which is the total genetic complement that every individual has, comprises approximately 3 billion base pairs of DNA. All of these packaged into small units in the nucleus of our cells called chromosomes. Now, each of our somatic cells in our body, not the sex cells, each of the somatic cells in our body contains 23 pairs of chromosomes. One pair we would have inherited from our mother, the other pair we would have inherited from our father, to give us a total of 46 chromosomes in every cell in our body. These chromosomes in the nucleus of a cell contain all of the information that a cell needs to carry on its life processes. The structure of the helix itself is comprised of 4 chemical bases known as adenine, thymine, guanine, and cytosine, or A, T, G, and C for short. The bases are shaped in a complementary fashion such that A will always bind along the double helix with a T, and G will always bind along the double helix with a C. And these twisted bases form the ladder of the helix as you look at that helical structure. So this DNA strand is, to stretch it out and look at it as though it's letters of the English alphabet, we've got the individual bases, the G, A, T, C, that make up the letters of the DNA. These letters are grouped into small groups of 3 or subsections of 3, where each group of 3 nucleotides makes what we call a codon, and each codon in turn links to a specific amino acid. So the genes create your codons, which in turn create your proteins, which perform all of the functions that are going to be necessary within the cell. So again, it's just shown in a graphic structure. We've got the DNA nucleotides, groups of 3 making up a codon. Each group of 3 makes a different amino acid. So we've got methionine recognizes the AUG codon, valine would recognize the GUC codon, and this is how the instructions are put together for proteins to get made. The amino acids join up with each other to form long stretches which ultimately make our proteins. Now, some of the terminology that's important to understand is this difference between genotype and phenotype. These words will be used a lot in the coming lecture series, and it's really important to understand what we're talking about when we talk about genotype versus phenotype. The genotype is the genetic makeup of an organism. It's the DNA that you're born with, the genetic blueprint that sits within your cells and that doesn't change through the course of your life. In contrast, the phenotype is the physical manifestation of the genotype. So it's the composite of an organism's observable characteristics or traits, such as its morphology, development, biochemical or physiological properties, phenology, behavior, and products of behavior. In other words, it is the characteristics that you see when you look at somebody, which are there as a result of the genes that they carry. Just to give an example, practically something we can all relate to is just looking at a dog, for example. At the top of the slide we see the genotype, which is the stretch of nucleotides that sits in the cell, the DNA material. We can see there that one strand has been inherited maternally, the other strand is inherited paternally, And together these form the basis of the characteristics that create the dog. The phenotype is what we see when we look at the dog. In this case, we've got a Labrador with its fluffy yellow fur, and that fur is as a result of the genes that it carries. If we were to change some of the letters in that dog's DNA, we would then get a chocolate-colored Labrador, and that would be changing the genotype, resulting in a change of phenotype. Much of what we know about genetics today is as a result of a major international collaboration started back in 1990 called the Human Genome Project. When it started, the goals of the project were to determine the sequence of these 3 billion chemical base pairs that make up human DNA. In other words, to read the book that comprises the genetics that make us human. And secondly, to identify and map the approximately 25,000 genes that we all carry within our DNA. The major outcomes of the Human Genome Project and the very basis of the work that we do within DNA Life was the realization that the DNA of any two individuals is as much as 99.9% identical. And furthermore, that the majority of the 0.1% variation has no functional significance. But crucially, there are small genetic variations within our DNA which can influence the ability of the genes to perform their functions. Now, when we talk about genetic changes, these can happen on two levels. There can be a genetic mutation or what we call a genetic variation. When we talk about a genetic mutation, it's a change in the genomic sequence of an organism that will cause a discernible change in the characteristics of the organism. I mentioned this as an example earlier where we changed the letters of the genes within the Labrador and it resulted in a change from yellow fur to brown fur. That would be a genetic mutation causing a discernible change in the physical characteristics of the organism. These are termed high penetrance, and when you carry the genetic variation, there's a very, very high chance that it will result in a physical manifestation of that variation, be it an appearance, a disease, some sort of modification or alteration in the biochemistry of an individual. Now these can be in the form of a single base pair. It can be an entire gene that is missing. It can be a large segment of a chromosome that is either missing or deleted, or it can be an entire chromosome that is missing or has been duplicated. So these are genetic mutations and they result in an effect that is expressed and that is visible. Another example would be the onset of sickle cell anemia. So what we're seeing in the slide is the change of a single nucleotide in the DNA sequence from a T to an A, and this results in the amino acid change from glutamic acid to valine. And just the simple genetic change at the DNA level results in a modification to the structure of the red cell membrane, resulting in the disease known as sickle cell anemia. This is a classic example of a genetic mutation. In contrast to the other end of the spectrum, we have what we call genetic variations. Now, we within DNA Life and all of the tests that we sell are far more concerned with the genetic variations, because here we're talking about low penetrance markers which by themselves are insufficient to cause a disease or cause a physical trait or characteristic, but rather they convey a susceptibility. That susceptibility, in combination with the environment, will determine whether the characteristic ultimately manifests. These are most often in the form of single nucleotide polymorphisms, or as they're more commonly known, SNPs. So a SNP is a gene variant that is caused by a change in a single nucleotide or base. And these are the tiny variations in our genome which make a significant difference by affecting the ability of our genes to perform their critical functions. In the same way as changing the letter of a word can have a major change in the meaning of the word, simply changing the P to F obviously changes the word quite drastically from pig to a fig. So exactly the same thing will happen at the DNA level, where the change in a single letter in our DNA can cause a change in the structure of the amino acid, as we saw with the sickle cell anemia example, which can affect the manner in which that gene is expressed. You can get either normal gene expression on the left, where you've got the nucleotide C, and if you change that C to a T, you've got the variant form of a gene. You have altered conformation or an altered structure to the protein that gets created, and this in some way will alter your gene expression. It can be an increased gene expression, it can be a reduced gene expression, but there's going to be some effect on the manner in which the protein is able to perform its required function within the body. Now, we spoke earlier about the 3 billion base pairs that make up our DNA, and that the genetic variation exists within less than 0.1% of this 3 billion base pairs. 0.1% of 3 billion is still a really big number. So how do we at DNA Life decide which of these SNPs are relevant for inclusion in our genetic test? And we've got some very specific criteria that we look at to justify including a SNP in the test for analysis and reporting to the doctors. First of all, the SNP needs to be relevant. It needs to exert direct influence over a specific biochemical process, and that biochemical process would ordinarily be known to create known symptoms of disease. Secondly, the SNP needs to be prevalent, so it needs to be common in the population. There's no point in looking for genes within our tests that we're simply never going to come across in the general population. Importantly, the SNP needs to be modifiable. We're talking here about the lifestyle or dietary or nutrition interventions that we are able to recommend on the back of these genetic tests. So the SNP expression is modifiable by environmental factors such as diet and lifestyle. We are not interested in giving information to either the practitioner or the patient where there's nothing that they can do to act on the information that we're giving. So every single gene that we look at, there is an intervention that is available to modify the effect of that SNP and essentially negate the effect of that SNP if that SNP is having a detrimental effect within the cell. And ideally, this is not always the case, but ideally you want the impact of those clinical interventions to be measurable through laboratory analysis. And this is where the test pairing concept of DNA Life becomes so relevant, because if we're looking at genetic tests together with functional tests, and we're using the functional tests to assess the responsiveness of the individual to the biochemical interventions or nutritional interventions that we're able to make, then we can see that what we're doing is being beneficial to the patient, and we can tangibly measure the outcome of these interventions. Just to end off this introductory lecture is to familiarize yourselves with some of the terminology that you're going to see in the reports in terms of the way the gene names are written and how we refer to the names of the variations that we find. So what we've got here is some of the terminology that relates to sequence variation within the DNA. You see that the gene name is written as CRP, which is an abbreviation I'm sure you're all familiar with, of C-reactive protein, 219GA. Now what this means, the CRP is the gene name. The 219 refers to the position of either the nucleotide or the amino acid on either the DNA or the protein strand on which it's found. And the G to the A is telling you the variation that we're looking at. So in this particular example, we know it's nucleotide because it's G and A. So at position 219 on the CRP gene, there's sometimes a change from a G to an A. What this means is that the CRP gene has two alleles. It has the G allele or the A allele, where an allele is defined as an alternative form of the same gene or the same genetic locus. So in this case, you've got your G allele, you've got your A allele, and this results in 3 possible genotypes, bearing in mind that you have 2 copies of each of these genes. You could have either the G being inherited from both father and mother, in which case you are known as GG. You could have the G from the father and the A from your mother, in which case you've got 1 copy of the most common form, the G, and 1 copy of the less common form, which is the A. In this case, you'd be written as GA, which would be called a heterozygote. And finally, you could be a carrier of both of the minor alleles, in this case the A, so you've got the variant on both chromosomes, in which case it's called homozygous. So in this case, you'd be homozygous for the variant A allele. Now, just to reinforce the terminology, the GG would be termed the wild type. GA, where you have one copy of each of the nucleotides, would be called the heterozygote. And AA, where you've inherited both copies of the variant, would be called a homozygote. These can be written for either the amino acid or the nucleotide. If it was for the amino acid equivalent, and this time we're looking at the ADRB2 gene, instead of writing the G's and the A's for the nucleotides, we now write the abbreviations for the amino acids. So in the ADRB2 example, we're looking at an ARG16 gly, which means at position 16, at amino acid number 16 in the protein, we're changing from arginine to glycine as a result of a nucleotide change. And in this case, it would be written as ARG-ARG, ARG-gly, which would be your heterozygote, or gly-gly, which would be your homozygote. We try and be consistent with the literature in terms of the nomenclature that we use in our reports. And sometimes it does refer to the nucleic acid, sometimes it refers to the amino acid. But either way, the concept of what we're trying to get across is that we're changing from the wild type or the most common form of the gene to the variant form, and we're showing you whether you're carrying one or two copies of the variant, and it's showing you at which position this variation is occurring. So hopefully now this has given you a better understanding of some of the basic concepts and terminology that are associated with the DNA life tests. You'll be learning a lot more in the coming lectures about the specific tests that we sell, as well as the biochemistry and molecular pathways behind these tests, as well as a lot of the genes that form these tests. So there'll be a lot more detail in the coming lecture series, but hopefully I've managed to equip you with some of the tools to better prepare you for those lectures going forward. Thank you very much.